Hello students, hope so you are doing well in your studies. Today our topic of discussion is nucleic acid, and nucleic acid, why it is so named? Because of their isolation from nucleus, and due to their acidic nature they are called as the nucleic acids. And first of all, they were isolated from the nucleus of the pus cell, and it comprised of two things, DNA and RNA. DNA means deoxyribonucleic acid and RNA means ribonucleic acid. It is very frequently asked question that the location of the DNA and the RNA. DNA is mainly found in chromosomes, nucleus, and in many organelles like mitochondria and chloroplast. If you forget about the organelles, which organelles contain the nucleic acid or DNA, you can remember which organelle is self-replicating. So there are only two organelles that are present in any cell that are self-replicating, that are the chloroplast and mitochondria. So they contains DNA, and RNA is present in the two things, major ribosomes and in nucleolus, and one is the cytosol. So let's start with the reading. Nucleic acid were first isolated in 1869 by F. Mister from nuclei of the pus cells. Due to their isolation from nuclei and their acidic nature, they were named nucleic acids. Nucleic acids are of two types, deoxyribonucleic acids or DNA and ribonucleic acid or RNA. DNA occurs in chromosomes in the nuclei of the cells and in much lesser amount in mitochondria and chloroplast. RNA is present in nucleolus, ribosomes, and in the cytosol, and in smaller amounts in the other parts of the cells. And the next question is very important. What is the difference between nucleotide and nucleoside? Actually, nucleotide and nucleoside, it is very easy concept, that if there are only two components, one is nitrogen's base and one is ribose or deoxyribose sugar, it is called the nucleoside. But if we add phosphoric acid to the nucleoside, it will become the nucleotide. So the nucleic acid are complex substances. They are polymers of the smaller units called nucleotides. What is mean by polymer? Actually, as we have discussed in the proteins, we were discussed amino acids are monomers and they are joined by the peptide bond to form a polypeptide chain. So the proteins were the polymer of amino acids. In the same way, nucleotide is the isomer and they are joined by the phosphodiester bond and they form a polynucleotide chain that is called DNA or RNA. So DNA is made up of deoxyribonucleotides and RNA is composed of ribonucleotides. Each nucleotide has three subunits: a 5-carbon ring monosaccharide, it may be the pentose sugar, and it may be the oxyribose or deoxyribose. You can say it may be the ribose or deoxyribose. Ribose is present in RNA while the deoxyribose is present in DNA. And what is meant by deoxy? It means from carbon number 2 of pentose sugar, if oxygen is removed, it is called the deoxy. Deoxy means removal of oxygen from carbon number 2 of the pentose sugar. So nucleotide contain pentose sugar, nitrogen's base, and phosphoric acid. Pentose sugar in nucleotide, ribonucleotide or RNA is ribose, and in the DNA it is deoxyribose. Nitrogen's bases are of two types. They may be single ringed or they may be double ringed. If they are single ringed, they are called pyrimidines, and if they are double ringed, these are called the purines. You can remember it by reversing the name. Pyrimidines contain di, so it is single ringed. Reverse, single ring contain word di, and purine is double ringed. And the pyrimidines are the tuck, tuck, and purines are ga, that is guanine and adenine, and the pyrimidines are single ring, that may be thymine, uracil, and cytosine. So actually thymine is present in only DNA and uracil is present in only RNA. Uracil is absent in DNA and thymine is absent in RNA. And the third component is the phosphoric acid has the ability to develop ester linkage with OH group of pentose sugar. In a typical nucleotide, in nitrogen's base is attached to carbon number 1 of pentose sugar, while phosphoric acid is attached to the carbon at 5 position. You can see it by diagrammatically. At the carbon 1 of the ribose, that is the nitrogen's base, adenine is the nitrogen's base, and carbon number 5 is attached to the phosphoric acid. So in the next we can discuss about the nucleoside. As we discussed, the nucleoside contain only two components. If we remove the phosphoric acid from nucleotide, the remaining part will be called the nucleoside. The compound formed by a combination of a base and a pentose sugar is called nucleoside. Nucleoside and phosphoric acid combine to form nucleotide. Each nucleotide of RNA contains ribose sugar, while sugar in each nucleotide of DNA is deoxyribose. Deoxy means, as we have discussed, one oxygen removed from OH group at carbon number 2. ATP is also an important nucleotide used as energy currency by the cell. Let's start with the DNA. Actually, the nucleotides are attached with the phosphodiester bonds. That is a major bond, that is important MCQ. And there are so many examples. You can see D-adenosine monophosphate, D-guanosine monophosphate, D-cytidine monophosphate, D-thymidine monophosphate. These are all 4 kinds of nucleotides. DNA is a hereditary material. It controls the properties and potential activities of the cell, and it contains all the genetic information. You can say it is the control center of a cell. It is made up of 4 kinds of nucleotides: D-adenosine monophosphate, DAMP, D-guanosine monophosphate, D-cytidine monophosphate, and D-thymidine monophosphate. These nucleotides are united with one another through phosphodiester linkage in a specific sequence to form long chains known as a polynucleotide chain. 2 nucleotides join together to form a dinucleotide, whereas if 3 nucleotides are joined together, they will call the trinucleotide. NAD, nicotinamide adenine dinucleotide. As the name represents, it is a dinucleotide and it is a coenzyme that is used in several oxidation reduction reactions in the cells. And in the next part, mostly these MCQ are required or asked in the papers. Name of the scientist and their functions. Actually, there are 3 main scientists that have different functions. Erwin Chargaff provide the data that adenine in the cell is equal to the thymidine if it is DNA, and if it is RNA, adenine is equal to the uracil, and the guanine is equal to the cytosine. Erwin Chargaff provided this data. And next is Maurice Wilkin and Rosalind Franklin used the X-ray technique, and Watson and Francis Crick built the scale model of the DNA. Actually, there are 2 bonds between adenine and thymine that is represented here. Adenine and thymine contains 2 bonds, and guanine and cytosine contains 3 bonds. So this is asked, mostly asked in papers, and 2 strands are wound around each other so that there are 10 base pairs. You can see the diagram here. In each round, here just 3 represented. Actually, there are 10 base pairs that are present in one turn, and the diameter is 34 angston. 2 strands are wound around each other so that there are 10 base pairs in each turn of the about 34 angston. And you can see the amount of DNA is fixed for particular species. It is very important, because if the amount of DNA will be not constant and or it may vary, then the new offspring will of course contain the double or triple amount of DNA. So it is very necessary. What is RNA? Actually, nitrogen's base is uracil instead of thymine, which is present in DNA. And actually I will discuss about you guys about the transcription and translation, which is also very important short question asked in papers and in NTS papers. Very important MCQ. Transcription and translation. Actually, the word transcription mean the formation of RNA from DNA. You can say that the formation of single stranded RNA from the double stranded DNA, it is called transcription. And what is translation? Actually, the translation is formation of protein from that RNA, and the whole process from the DNA, the formation of RNA, and from the RNA formation of protein, the whole process is called the central dogma. The whole process is called central dogma. So the central dogma is transcription plus translation. The whole process is called central dogma. The next is the types of RNA. That is very important long questions, and is very important MCQ that are frequently asked in MDCAT. There are three types of RNA: messenger RNA, transfer RNA, and ribosomal RNA. As the name represents, messenger RNA, it carries message; transfer RNA, it transfers something. We will discuss in later. And ribosomal RNA, due to their location, they are called the ribosomal RNA. Are these three types of RNAs are synthesized from DNA in the nucleus, and then are moved out in the cytoplasm to perform their specific functions with the help of the nuclear pores. We will discuss in chapter 4 what are the nuclear pores when we will discuss about the structure of nucleus. Messenger RNA, the major thing is that first of all you should know it is about only 3 to 4% messenger RNA, and transfer RNA, it is just 10 to 20% of the total RNA in the cell, and ribosomal is 80%. You can say ribosomal RNA is most abundant, then less amount is present in the transfer, and then messenger RNA. After that, it is very important, messenger RNA is a single strand of variable length. Messenger RNA depends upon the structure of the protein for which it is taking the message. Actually, it takes the message from the nucleus to the ribosomes, and then ribosomes aggregate to form the polysome, and the structure of new RNA is formed, and new proteins are ultimately formed. Actually, as the name indicates, it takes the genetic message from the nucleus to ribosomes in the cytoplasm to form particular protein. Messenger RNA carries the genetic information from DNA to ribosomes, where amino acids are arranged according to information in messenger RNA to form specific protein molecule. This type of RNA consists of single stranded of variable length. Its length depends upon the size of gene as well as the protein for which it is taking the message. Actually, the protein size matters. For example, if we have to synthesize a protein of 100 amino acids, then of course we will have the messenger RNA for 300 nucleotides. For example, for protein molecule of 100 amino acids, messenger RNA will have the length of 300 nucleotides, and as we discussed, messenger RNA is about 3 to 4%, least frequent is present. And the transfer RNA, the name represents, it transfers the amino acids actually, where the site where peptide bonds are being synthesized. Actually, it deposit the amino acids to the site where peptide bonds are formed, and then the proteins are formed ultimately. And it has a fixed length of 75 to 90 nucleotides. And there is one special feature, is that there is one specific transfer RNA for each amino acid. As we discussed in the protein section, most of the proteins are found by 20 types of the amino acids, so there will be 20 types of transfer RNA in present for that requirements. It comprises about 10 to 20% of the cellular RNA transfer. RNA molecule are small, each with the chain length 75 to 90 nucleotides. It transfer amino acid molecule to site where peptide chains are being synthesized. There is one specific transfer RNA for each amino acids, so cell will have at least 20 kinds of transfer RNA. Transfer RNA picks amino acids and transfer them to the ribosomes, where they are linked to each other to form the proteins. Actually, on the surface of the ribosome, it transfers the amino acid for the formation of proteins. And the next is the ribosomal RNA. It is a major portion of RNA in the cell and may be up to 80%, and it is strongly associated with the ribosome, as the name represents 40 to 50% of it is present. It acts as a machinery for synthesis of proteins. And the next line is actually the main thing. On the surface of ribosomes, the messenger RNA and transfer RNA molecule interacts to translate the information from genes into specific protein. Actually, on the surface of ribosome, the messenger RNA and transfer RNA molecules interact and form specific proteins. Thank you very much. This was all about the nucleic acid, that is DNA and RNA. First of all, we have discussed about the DNA, in which we discussed the difference of nucleotide and nucleoside, and then we discuss about the thymine, that is present in only DNA, and the uracil, that is present only in RNA. Then we have discussed about the pyrimidines and purines, that are double ringed and single ringed bases. And after that we have discussed about the RNA transcription, translation, and the types of RNA, that are the messenger RNA, transfer RNA, and ribosomal RNA. Ribosomal RNA is maximum, and after that transfer RNA, and after that messenger RNA. Hope so guys you understood well, and if you gain some knowledge from this video, you can subscribe my channel. Thank you very much.